Does it work?
Welcome everybody to the morning session. We will start the first talk by Benoit Perthame, who is going to talk about bacterial movement by run-and-tumble. Please, thank you.
So thank you very much. And thank you very much for the opportunity to be here. So what I understood from the talks to the beginning of the week is that questions of biology are not really in the heart of this community. So I assumed that people do not know about bacteria. And so I will give more an history of the subject, why kinetic equations are used in this field. And so I'm sorry for the few experts in the room who already know all this, but it will be really an history of the subject since 20 years. Okay, so that's the first problem. For some reason, it doesn't want to switch with this. Okay, I'll do it with my hand. So to give a path to the talk will be to explain why to describe the motion of bacteria, at some point you need what I call the flux limited Keller-Segal equation, which is this Fokker-Planck equation, where you have here n density of cells who are moving with a random motion, and we have a drift. And this drift is the gradient of a chemoattractant or a food or whatever, of a signal, let's say, which is related to the solution itself, to the cell density. It's emitted. If in the chemoattractant case, the cells emit this concentration of chemoattractant. And the reason why it's called flux emitted is that there is a saturation by the gradient C of this drift, which means that you have a nonlinearity in this right-hand side, which you find in many equations. It's not only there that you find this kind of, it comes from geometry, it comes from image processing, many places. So there have been many people working on that. This is a list of people who have been working on these kind of things. So because of the flux limitation, the drift is bounded, which means that existence is not really a problem. It's, you have easily uniform bounds on the solutions. So the real question for this equation is not a question of existence, uniqueness, or regularity. It's more where it comes from. Why are we interested in this? So this is the path of the talk. I will first tell you why we do that, why the world of bacteria is fascinating. Then I will give you a node kinetic model which represents these kind of things. I will tell you how you derive the flux limited Keller-Segal equation from a kinetic equation. I will tell you about properties of the solutions, and interesting properties are instabilities. These equations have instabilities, which is why they're interesting. And as you will see, the kinetic model is not very nice. And I will give you, explain you where it comes from if you want to be more precise in terms of biology. So these are the five sections. So first of all, why are bacteria interesting? Bacteria are interesting because we live with bacteria in our body. We eat food produced by bacteria. Bacteria are very bad in hospitals. They can create disease. So they're very important. And they have properties which is, even though they are very simple, that more or less the first organisms in life on Earth. This bacteria has a capacity to move collectively in a very surprising way. And these are colonies of bacteria which have been initiated by just an inoculum in the middle of this dish. And so they divide, they move, and they have learned how to invade a petri dish, a domain with this kind of shapes. So they have ways to communicate in such a way that they can find food in much more efficient way as if they had a spherical wave. So you see this kind of equation of patterns. It's been maybe 40 years since we have mathematical models which represent that. And this is an example, if I can get rid of this window. This is an example of what you see in a parabolic equation. This is the solution of a parabolic system. Just to tell you that the parabolic system can be very, very strange. These are solutions of parabolic equations, which is a model to late Professor Mimura, who proposed this equation for this parabolic equation for this. So these kind of things so these kind of patterns are quite interesting. These bacteria are close to Salmonella, those you find in food. They just move because they try to find nutrients, and those which are in advance, they have an advantage. So they eat the food, and those who are behind, they have no food left. And so because of that, they have an advantage, and so you create an instability. So this is the first kind of objects you find in bacteria. What we are going to be interested in is not at all this. It's another phenomena, which is called chemotaxis, and which is the way to communicate between bacteria through a molecule which they like, and so they are attracted by the molecules they produce. So in the 60s, Adler made a very famous experiment where he had bacteria at the end of this kind of tube, and he opened the tube. Bacteria are moving together, and what he observed is that they move like a traveling band. So what you see in red here is the density of cells, and you have a peak along this line in time. So they are really moving with a constant speed. So how can you imagine that bacteria, which are very simple organisms, no eyes, no brain, nothing, they just move because they have flagella to move, they swim, and they communicate by emitting chemicals and reacting to the chemicals. And this is the result of this communication. So these experiments have been done 10 years ago, again by Curie Institute. And it turns out that if you want to explain them, okay, I can show you, by the way, the movie showing the wave moving. So this is the wave. It's initiating on the left. Sorry about that. It's initiating, moving like that until it reaches the end of the tube. So this is what you see in these experiments. Oops. Okay. I'm lost. Yes. So this is what you see in this experiment. And what we discovered with this group and Vincent Calvez and this group of experimentalists is that if you want to explain that, the flex-limited Keller-Segal equation is the correct interpretation. So this is what you observed. So you take the cell density, you take a velocity, a drift, which is due to two things: chemoattractant. This chemoattractant means that cells want to stay together. This is the first field. And then there is a nutrient, a substrate, and cells lack the food, they consume the food. So like in the first experiment, in the first movie I showed, they move where the food is. So this one, the food is used by the cells, is depleted. So the cells will try to find the food further on the right. And this keeps the cells together. So you see that because of that, cells want to stay together and they move from left to right. So there are two effects, which are both reaction to chemical molecules like that. What is very surprising is that you have an asymmetry of the pulse. You have a long tail here and a sharp front in here, and that the model, the flux-limited Keller-Segal equation, is able to reproduce that precisely. Okay, so this is the reason why we are interested in this equation. It's because it fits perfectly with the experiments, which is not so often in biology. So after this motivation, I will come now to the kinetic model, which is behind. So what we would like to explain now is this flux-limited Keller-Segal equation, but not departing from the Fokker-Planck equation, by explaining it by the individual behaviors of cells. So this is known since the 80s that bacteria are moving by a run-and-tumble, as it was mentioned by Christian yesterday, which means, so cells, this is Escherichia coli. You have these flagella, they move with these flagella, they use them as a propeller, they move, and so they make long jumps, and sometimes they change direction, make another jump, change direction, make another jump. So this is exactly scattering. So they move like that. It's called run-and-tumble. The effect of run-and-tumble is, for example, that you can observe patches of cells where cells are moving exactly with run-and-tumble like that. They are attracted by the center because they are chemotaxis, so they lack the food, which is, or the chemical which is emitted by the cells. So they are attracted in the middle, but they have a memory which makes they don't react exactly as fast immediately. So they move, and time to react, they come back and then come back. And this was studied by very much by Christian and Vincent Calvez, and you have explanation on that. So this is the way it works. So when you follow bacteria, what you follow are this kind of path, which is a succession of run-and-tumbles with a drift, which is following the colony of bacteria. So the main density of bacteria is here, and cells are moving, but more or less they follow this direction. I'll show you the movie, which is a way to visualize what is multiscale analysis, because you have some cells, some of them which have been engineered to be visible, and if you follow them, they do run-and-tumble. If you see the global effect of the run-and-tumble, it's simple. You have a band here in the middle. If you have good eyes, you have a band. And during the time of the movie, the band moves a little bit from the left to the right, from the center to the right. Okay, so I show you again this. You see, so what you see here in the middle is this colony of cells, and you see that globally, macroscopically, it moves to the right. Okay, So this is what mathematicians call epsilon going to 0. You have a random motion of these cells, and behind that there is a macroscopic effect, which is this light band moving. So to explain that, in the 80s, a group of people around Alt, Dunbar, Otmer proposed a kinetic equation, because you have a run and tumble. So cells have a velocity which are called psi. They have a position which is x, and they have a run and they have a tumble. And all the difficulty, all the modeling problem is in the tumbling. So tumbling means that a cell of velocity psi prime will turn with a certain rate to a velocity psi. And of course, all the cells with velocity psi prime have to be subtracted from this balance. So here I write the model only with a single molecular content, C, assuming it is given. In practice, for what we are interested in, there would be a second one. There would be also a substrate. And these are not independent. They are connected to the cell density, so the integral of f d psi by diffusion equation. But for the time being, let's just look at this equation. Here, the velocity space, I call it B. It could be either the during the jump, the cells have always the same speed. So in some sense, the velocity space should be the sphere. It's not true, because in fact it is really a 3D experiment. You have a 2D tube that you have seen, but there is a thickness. So there are cells which are moving also up down. So which means that it's reasonable also to say that they are not only on the sphere, but on the ball, because those are going exactly up and down at velocity 0 if you take a 2D experiment. So this is the velocity space. And of course, this can be used to derive macroscopic equation from that. So to do macroscopic limit, you need a small parameter. You need to know what is the scale which you are going to use to rescale. Here, if you think of the simplistic model, what you could say is that cells, they are making these jumps. During the jump, they measure the concentration of chemoattractant. So this molecular concentration C, they measure it along the path. And so if you take an average middle point rule, this means that you have a parameter epsilon here, which is a time scale connecting, which tells you, connecting the jump to the velocity and telling you what is the concentration they consider. So you could say, okay, cells along the path, they have measured the concentration, and depending on this concentration, they decide to make a jump, to tumble or not. So if you do that, you have your small parameter. You can use the standard machinery. You rescale. You do a diffusion scale. You create this diffusion limit, and after working a little bit, what you find is that the solution f epsilon will lose its dependency in the velocity. So the distribution will be uniform on the ball, on the sphere, and that the macroscopic quantity satisfies the Keller-Segal equation. But it satisfies the Keller-Segal equation with coefficients depending on your decision of what is a K. So what is a kernel? So the diffusion coefficient is proportional, inversely proportional to the measurement of the concentration K, but the sensitivity here, the drift, is proportional to the derivative of K. So it's only simply that when you have a collision kernel, the symmetric part and the asymmetric part play different roles. One of them plays on the drift, the other one plays on the diffusion. And that's what you observe here in this particular case. But it's not flux emitted. It's just maybe limited, but only in C, but it's not limited in grad C. So it's not the correct interpretation for what you want to do. It's not a good model. Especially because the Keller-Segal equation here, it's well known, blows up infinite time. So if I connect that with a concentration C emitted by the cells, like I showed in the first transparency, you say that cells emit the concentration, then the solution of that will blow up infinite time. There is concentration of the cells. All the cells come together as a Dirac mass at some points, infinite time. So this means that the result is for short times just because it's of this blow-up phenomena. But in any case, we don't care. It's not the correct model. So the real equation, kinetic equation for the flux-emitted Keller-Segal equation is a little bit different. It has the same flavor, but it is more accurate in terms of biology. And that's what I explain now in this section. So what happens, what do cells do? They move, they run and tumble. But they decide that when they feel that the concentration C is increasing, they will have longer path. If they go in the bad direction, where the chemoattractant diminishes, they don't like it and they run, and they turn very quickly. So they will make long runs where the chemoattractant is increasing. They will make short path, short jumps when the chemoattractant is decreasing, which means that they measure not the concentration, which of course, if you think a little bit of it, is a little bit absurd. How can a cell decide what is the concentration or not? What a cell can do is measure a difference of concentration. Because according to time, it can say if yes or not, the concentration increases or not. So that is what is the real dynamics of the cells, of the bacteria. So if you put it in the equation, now you find, and this was proposed by Yasmin Dolak and Christian Schmeiser some time ago, what you do is you say that your kernel K depends on C, but not on the values of C, on the path derivative of C. So you measure if the concentration increases or not along the path. So the path, the concentration along the path is varying like that. And so this is the way it works. In particular, in this picture, a cell can only decide of concentrations along the path, along their velocity. They have no way to decide of the new velocity they will take. It will be uniform, which is questionable in terms of observations. So the very stiff example is capital K here, bold K takes two values. If this is negative, cells do not like it. They jump with a parameter K minus, which is very big because they jump very quickly. They don't like to go in that direction. If this concentration increasing along the path, then DTC, strictly positive, and though they jump with a red K plus, which is small, because they like it, so they continue in the same direction. So if you take this very stiff case, this discontinuous case, which is an extreme case, then what Dolak and Schmeiser proved is that because you depend only on psi prime, you have an equilibrium you have an equilibrium and you have an hyperbolic limit. You don't need to go to the diffusion limit. The average will already converge to a singular hyperbolic equation. You need just an equation for the first order, and the coefficient will be very singular because the coefficient will depend on grad C divided by the modulus of grad C. And of course, solving a transport equation with such a bad object is a complicated thing, and all the work was done by François Jam and Nicolas Vauchelet, and around ideas of very weak flows that you get by duality. There is another point of view which is more standard, which is to make a diffusion limit. So if you want to do that, what you say now is that your K epsilon is not that stiff. It is smooth, but stiff. And epsilon is a stiffness parameter. So maybe I can draw a picture if I can find chalk here. So in some sense, if I put here DTC, rather than going from this to this, which is a stiff case, what you do is you do something which is like that. This is 0, and this is your parameter, which I called epsilon. And of course, because it's blue, you don't see anything. So let me draw. Okay, that's the epsilon. So this capital K now will be smooth. And you can use this parameter epsilon to rescale the equation in the diffusion limit. And if you do that, and I will not go further on that, what you obtain is a flux-limited Keller-Segal equation, where the phi now depends on the K by a very simple rule, and it is smooth. So it is exactly in the form of a smooth function, which decreases like 1 over grad C when grad C is large. So it's limiting, but in a smooth way, the flux. So with this kinetic equation, you recover really the flux-limited Keller-Segal equation with this procedure. So at this stage, I will maybe stop to speak of a different question, which are instabilities. So at this stage, are there questions? Is everything clear on the motivation and how we arrive to the equation? Yes.
Concerning the flux limitation function.
Yes.
There is unique formula or there are parameters?
No, you take any function like that, any one, and you put a parameter epsilon to rescale it, and then you get it. You get a formula. There is nothing very complicated. No, it's so yes. Oh. What you mean, a collision of cells?
No. So, for instance, they come in different ways, right? We talk about, mostly I think we talk about E. coli.
Yes. No. Okay, no. So they are swimming in the fluid, but these people I mentioned at the beginning who did the experiment measure precisely. In that case, hydrodynamic doesn't play a role. They are too small. These are not the big enough cells. So there is not this interaction between the fluid and the cells that you find in bigger cells, or in some cases here you can find it. But in these experiments you can neglect, and it is measured that you can neglect. So instabilities. So instabilities is a magic word in biology because life doesn't like uniform states. So all these questions of getting entropies, uniform distribution, equilibrium, in some sense, never occurs in biology. I mean, nature hates things which are uniform. They like patterns. Whenever you look at anything created by nature, landscapes, animals, they have patterns. And the reason of that is very simple. If you have an organism which creates, like human being can do very easily, very beautiful fields which are completely uniform, if any parasite come, it will destroy everything. So nature knows that since for billions of years. this is all those who are uniform disappeared for some reason, and the life is completely unstable. And this model, both the kinetic and the macroscopic, has this nice property that they create instabilities, which means that if you, for example, create a Fisher-KPP type growth and say that cells are dividing, then what you observe is that the solution will not be uniform. It will have, it will exhibit oscillations. So this is a calculation done by Shugo Yasuda by a Monte Carlo code. At the beginning, of course, doing a Monte Carlo code, you see fluctuations. Okay, you see it's normal, there's nothing surprising. But at some point, it discovered that they are really too regular to be really random. And so he put a number of particles which is so big that no experiments in biology will do the same with the number of cells. But with this number of particles, he could get very precise Monte Carlo solutions, which are not fluctuating, but which exhibit this instability. If you look at the flux-limited Keller-Segel equation, again with a growth term, again you get this instability which you measure. And in that case, you can get either propagating waves, but because there is chemoattractant, you can have also waves which are backward propagating because they are attracted by the back, and so they go back. But they are always, and these are not highly, this is because it's a long domain. So these are patterns. It's not oscillations, high frequency oscillations, it's really patterns. And so you can analyze that by the usual linear instability. And what you can observe is that indeed, when epsilon is small enough in the kinetic equation, or if you look at the flux-limited Keller-Segel equation, then the state n equal 1, which is the stable state, in fact, is linearly unstable. And it is unstable in the sense of Turing, which means that the frequencies, the wavelength, if you want, are always finite. They never go to zero. The wavelength doesn't go to zero. The frequencies remain bounded. So in some sense, I will stop here. So you go to that, you find the dispersion relation, which should be here. This is a dispersion relation. So this quantity has to create an instability. So lambda is the exponential growth in time. So you need this number to be positive, and you see that the frequency which work cannot be too large. If they are too large, the diffusion is winning because this is a diffusion term. Diffusion is winning. But if the model is unstable, which in this calculation means phi of 0 is big enough, then there are values of k for which this number is positive. So you get linear instability, but in the Turing sense, which means that the modes are bounded. And so you have patterns according to the Turing theory. Okay, so the conclusion is that we are happy. The flux-limited Keller-Segel equation represents well the data, fits well the data which we have. It has this beautiful instability property. And the only thing which is a little bit weak is that the kinetic model which I use is very strange. So during the week, we have seen this Boltzmann equation where geometry is so nice. We have seen Vlasov-Maxwell where you have all this beautiful structure. This kinetic model is awful. It has to measure the variation of a quantity along a path and decide of the scattering according to that. There is no nice geometric structure. So you don't like it. So in fact, there is an explanation for that. And if you want to find an explanation for that, you have to go to another scale, which is the size, the scale of the cell. So the cell has a membrane, which is here. Outside, of course, this membrane, there are what are called transmembrane proteins, receptors. which have a head outside and head inside, and they measure the concentration by this head outside. So molecules can come and attach to these receptors. By the way, we heard that every week about COVID, that the way virus attach to the cells. They are receptors and they attach to the receptors. And in our case, outside you have a concentration C of chemoattractant. These molecules can attach or not to the receptor, and then when they attach, they produce a chemical pathway inside the cells, and if they detach, they create another pathway. And all these things behind are just molecules, and at the end, you have a molecular motor acting on the flagella. So this is how cells are moving and are reacting to the external world. And so this is clockwise and this is counterclockwise, and this depends on tumble or run. So this is what happens in practice. So if you want to take this into account, what you do is to introduce another variable. So you introduce an internal variable which tells you what is the state of the cells inside, okay, compared to the external concentration of molecule. Okay, so this quantity, some people use methylation level. Some other values are possible. So this is an internal state of the cells. So I know you told me during all the week that to solve Boltzmann equation is more or less impossible because you are 6D now. But the good news is that only 2D. Basically, all this is always done in 2D. So, but we have one more variable. So we have this new unknown, which is this new variable m, which is the internal state, and we have this function f now, which depends also on m. So what you write is a kinetic equation, Vlasov type equation, Vlasov and scattering, where now the Vlasov term is more or less a reaction term, which tells you how the methylation will change by a chemical reaction. So you simply write here a chemical reaction m dot equal R of M and C. So depending on the external signal, the internal variable, internal concentration will adapt to the external signal to get an equilibrium because it's a chemical reaction. And on the right, you have the tumbling effect. So the tumbling effect is that depending on the value of M and C and psi prime, we will decide to tumble or not. So how is it connected to this model that I call the awful model, which has no geometric structure? That's very simple. That's very simple. If so, you have to introduce the parameters at two places. So you have to say that the chemical reaction is very fast. So compared to the motion of the cell, you say that the methylation, the internal state goes to equilibrium very quickly. So this is the time scale. And then you have to say that the tumbling rate is stiff compared to the equilibrium. So this chemical reaction will bring M to an equilibrium capital M, which depends on the external signal. Depending on if you are far away from M or not, but with a very stiff response, you decide to tumble or not. So this will take the value K minus and K plus that we had here, depending on this disequilibrium. Okay, so this is the way you model what I explained on the cells at the beginning. Okay, so the theorem tells you that when epsilon could converge to 0 in this model with internal variable, then the f epsilon will converge to a function f bar, which only depends on t and x. The Dirac mass at equilibrium. So in this fast reaction limit, you are always at equilibrium, but the small disequilibrium remains and create exactly the kernel K for the function F bar. So departing from that, if you make this asymptotic analysis, what you recover is a kinetic equation with this kernel. So this kernel is not so stupid as it seems to be. It comes from this structure, which is quite natural. Then you have nothing to say. It's just quantity, which are scalar and which act as molecules act in this kind of phenomena. Okay, so maybe what time is it? So, okay, if we want some time for questions, so maybe it's not necessary to go into. So the only the reason, okay. So the mysterious thing which have to be explained is how a function K of M can create a function K of a dt of the path derivative. How come in this framework that this path derivative can come out of that? And simply the reason is that you have to rescale. If you want to prove that, you have to introduce a new variable, which is this variable here. You replace the methylation with the internal variable M by a new one, which is called Y, which is a disequilibrium once renormalized. And if you do that, of course, and you change variables, you see that you will get a derivative, path derivative of M, which will come somewhere. Okay, and this is the reason why you generate a term like the DTM, which now you see here in this algebra. Okay. Okay, so this is my last slide. So is this something you can observe experimentally or not? Does it come from only the mind of mathematician? No, it is not. It is something that in physics lab they can measure. So how do they measure time scale of this chemical reaction? Simply, they impose a chemoattractant which is varying in time. So if the time scale is long enough, so this means that the variation of the chemoattractant is slow, then you have this chemoconcentration, and the cells follow exactly the chemoattractant. So they turn exactly according to the variation of chemoattractant. If you are going too fast, then they have no time to react, and this becomes chaotic. This is what you observe here, which means that the times, they can find the time scale of a chemical reaction with this kind of methods. And here are some comparisons between numerics and experiments. The bad news is that the time scale are not correct. So now that I have finished my talk, I can tell you this is not the correct point of view. This is not the correct epsilon. It's something different. I don't know what. But this is not. This is a beautiful picture, but the time scale are not coherent with measurements. There are so many cells that maybe some cells do that, but not all of them. Okay. I will stop. Okay. I have another observation, but I will stop. I don't have to. I don't want to go in that direction. Since the famous paper of the 3M, what I call the M3 paper, Moe, Michler, and that we know that kinetic equations are related to fractional Laplacian and fast diffusion. Biology and cell motion is full, for those who like fractional Laplacian and Levy jumps, biology is full of this kind of observations that cells make very long jumps sometimes, and they move more by Levy walks than by a Brownian motion. So this is one of the papers, but you can find many of them, not only bacteria, by the way, cells in your body also move by Levy walks. So we could explain. So here the challenge is to explain it by the internal variable. How the action of an internal variable can explain this fractional Laplacian, and this is something we did with Veran some time ago. So you can do it. You can find scales to do that. Okay, so this is the conclusion. So this world of bacteria is fascinating. The flux emitted Keller-Segal equation is a good model for explaining these traveling bands, which were observed from the 60s. And all this relies on a run-and-tumble Boltzmann type equation. And the biology now is really to understand the molecules, how the molecules inside the cells play a role in all this, what are the pathways. This is the modern observations. So all this I told you is a long story where Christian was involved from the beginning with Peter Markovich a long time ago. The experiments I mentioned them are due to this group of people in the Curie Institute in Paris, Min Tang in Shanghai, Nicolas Vauchelet and Zian Wang in Hong Kong also worked very much on that. And the last results are with Shugo Yasuda and Veran. So I stop here. Thank you very much for your attention.
Any questions also in the chat, people joining online?
Not yet.
Not yet.
Thank you. Yeah, thank you for this very clear introduction. So I would very much like to take up the challenge of solving the even higher dimensional problems. but sort of the question that I would have in that context is, do you, in these systems, do you really see kinetic effects? Or, I mean, from a numerical point of view, is it more reasonable to go to the fluid limit and then just to solve the fluid equation? So is there something that, I mean, from a fundamental perspective, of course, you want to do the kinetics because it derives the equations, right? But is there some genuine kinetic effects that you see here?
Okay, very good question. I will not answer your question. I will answer to a related question. Is that I said this is not the correct scale. It means that there is another variable which is at equilibrium, and that the macroscopic equation will not be the one in T and X, but certainly Tx and the internal variable. So this is, I think, what answers your question. Is the macroscopic equation is certainly not correct, and if you want to be more accurate, you need to keep you have to get rid of the velocity because you want to get out of the equilibrium, but you need to keep an internal variable in the macroscopic equation. And I don't know if somebody did it. Maybe you know, Christian? No, nobody, I think, worked on that so far. But so the answer is that the macroscopic equation is not good. It is good to explain roughly this model, but it's itself an equilibrium of another equation which would be the correct one in terms of scale, the time scale that is observed.
I have two questions. Number one is you said that this internal state jump at a higher speed or faster speed. Is it experimentally confirmed how big this epsilon is?
So I said the response is stiff, which I think is correct, and I said the reaction is wrong, and I think this is not correct. The reaction of the cells, the time scale for cells to get to equilibrium compared to the external signal is not that fast.
Okay.
But the stiff response is correct.
So
the epsilon is not the same at both places. I put the epsilon at the same, two epsilon at the same epsilon. It's not the same epsilon.
I see. Okay. The second question I have is it convention that this concentration enters only through the collision operator? Is it possible to enter through some kind of acceleration term?
To under what? To under?
So the concentration C enters through the tumbling kernel?
Yes, and the reaction, the chemical
right,
reaction, yes.
Right. Is it possible to include C in some kind of acceleration term?
Oh, yes, yes. you could imagine that the speed of jumps depend on some molecules on the environment of the cells. So you could do that, yes. So but that opens a completely different game. It's a completely different part of biology. It's some cells diffuse, but the intensity of the motion, of the Brownian motion, depends on the environment. Okay, so if you think so, this is well observed in embryos that the cells create concentration due to the variation of the coefficient in the diffusion coefficient. So this can be possible, yes, and this is very active now. There are many observations on that, and many people working on that.
People are this spring type in the US. I guess you can create that by bifurcation. You always have a constant steady state there. Is that true? And the parameter
I don't know if delta if the parameter is if the stiffness is is very big. I don't know if you can get it. I don't know. What we did is only analyze the small parameter to get instability, but I guess you are correct. I guess there is a stable regime when epsilon here is very big. I guess you are correct, and there is a bifurcation, but I didn't work on that.
In the experiments, you showed the profile of the wave is not symmetric. So my question is it possible to observe that on the numerical simulation on the macroscopic model?
But the waves are not symmetric. It's
so yeah, but so is it possible to prove that on the
Yes. You got here the pink is the big line, which is a solution of the equation, and the blue one is the experiment.
Yeah. So
and these are the experiments.
And proof.
Mm-hmm.
Is it possible to have a I don't know
to prove that this
that
is the solution? Oh yes, I think it's possible. It's because yes, it's I don't remember well, but where you see it, but you see it very clearly here somewhere. I don't remember, but yes, it's an observation which is. Will you explain it completely from the model? It's very easy to explain it from the equation. I don't remember why, but.
Just me. You mentioned in the beginning
yes.
nutrient-driven instabilities.
Yes.
That was studied by Nimura for the parabolic system.
Yes.
Can you study them? Do you even have them with the kinetic model? When you change the nutrient and it changes the pattern, because in the parabolic case, it's a case. You change nutrients, you go from like radial to hexagonal patterns, or you have different ones.
Good question. Good question. Do we? Is there any experiment with nutrients at the kinetic level? I think I've never seen that. I've never seen that. I've seen models. We agree. We are speaking of different physics. Here, this is not the instability of chemoattractant. It's another physics, and that I've never seen. No, I've never seen. I've seen many numerics on kinetic equation, but with the traveling bands, but not on that. So I don't know.
Because also the food acts as a chemotaxis, no?
The food has a, yes, absolutely, especially, yes, that's what I use. But here it's not used as a chemoattractant. It's really the depletion of food. So that's really an effect, a physical effect. I mean, the cells eat the food is diffused. So around them, there is a depletion. So all around, there is no food for the others. And so those are late, have no food. So they will be even more late. That's a bad, yeah, the word is terrible. But,
Hello? Okay, good. So I couldn't really hear the previous question, so I apologize if I'm asking the same thing. So this is a result of growth, right?
this is the result of growth and motion.
Growth and motion. So your model will capture the growth pattern as well?
Yes, the model of Mimura contains a growth term depending on the food available.
I see, okay.
So yeah, that's it.
I see, okay, thank you.
But the motion is independent of the food. That they move all the same way, but when they are in front, they reproduce more. So there will be more cells and they will eat more food. That's the process. It's debated. It's not also it's debated in biology because some cells do not do that.
Okay, any more questions? No, okay, let's thank the speaker again. Thank you.